Okay, so we're ready to start. Welcome back, everyone. This is the course on bioethical issues at the beginning of human life in the Master of Science in Bioethics. So welcome back. As always, we'll begin with a little prayer. Oh, let me turn off the lights on the, so the projector can be seen better as the eyes adjust. In the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you for all your many blessings, for the gift of life, and for having us back in class today for the Christmas break that we had. We pray now that this new semester may go well for all the students and teachers here at St. Thomas University and throughout the entire world. We always pray for those who are in most need of your divine mercy, and particularly on this weekend where we have the commemoration of the legalization of abortion in the United States since 1973. We pray for an end to all abortions, that all mothers may realize the beauty and the gift of their child, even as challenging as it may be. We ask this in the name of Christ our Lord. Amen. Father, Son, Holy Spirit. Okay, did anyone see on the news anything about the March for Life in Washington, D.C.? You saw some? Yes. You remember what channels it was?
I watched.
Oh, okay. Then they had the full coverage, I'm sure. Yes. About any of the secular media, secular news that anyone see? No, I saw a little blurb on CNN very quickly, but then they focus on the controversy of Mrs. Pence.
Or attention,
to try
anything to distract.
Exactly.
And they'll focus on the March today.
Right, exactly. So you see, that's what we have to deal with. Okay, the secular environment that is not very open or amenable to these issues at the beginning of human life, at the end of human life. And that's why I always try to present the arguments of nature, Okay, so to summarize a little bit from last semester, what we did last semester was essentially a theological anthropology in the course on the basic principles of Catholic bioethics, looking at the origin of the human species as a whole, right? To realize that we may indeed accept the process of evolution as a mechanical, biological process that occurs on all living things without having to reject our theological beliefs in God. Excuse me, sorry. Without having to reject the belief in God and God as creator of the universe and creator of each one of us. So that was the intent of the course, to do this phylogenetics, to find our origins within the origin of all species on that evolutionary tree. And we see that we are one of the most recent species, biologically speaking, that have emerged, right? The most ancient species that we can tell, that we can infer from some of the fossil record, but mostly because of the amount of oxygen that there is in the atmosphere today, the most primitive or earliest species were those bacteria, and particularly the cyanobacteria, the bacteria that could photosynthesize and literally change the atmosphere from being very toxic, like methane and high hydrogen and so forth, to mostly the oxygen that we can breathe today, okay? So that, in contrast to the human species with all our complexity, physiological, psychologically, and even spiritual. And I find it interesting that that happened in a range on this planet in a range of about 3.5 billion years, right? If you recall, the Earth itself, the planet, coalesced about 4.5 billion years ago. And it took about, well, actually less than 1 billion years for those bacteria to emerge. Of course, the Earth had to cool down enough to sustain first liquid water and then life in itself, all right? All right, so in 3.5 billion years, we have a divergence from very early primitive organisms to the great variety of species that we have today. And I mentioned briefly that we have classified about 2 million species on Earth today with a scientific taxonomy, right, as distinct species, and then varieties within the species. But it's estimated that there are between 10 to 20 million species, so we've only identified maybe 5 to 10% of the species that actually exist. Now, where is that other 95% of species? What's your intuition? First, they're typically microscopic, right? So we don't see them at plain sight. We've discovered most of the species at plain sight. Every now and then you hear of a report of a new insect that was found deep in the jungle of somewhere, okay? But most of the visible species we've identified, typically they're microscopic and they're in the ocean deep, in the ocean bottom, all right, on the ocean bottom, where also, as you can imagine, everything that dies in the ocean falls down like constant rain, all the way from microscopic plankton to huge whales. Everything in there dies eventually, or is eaten up, and then the one who ate up whatever else also dies, and all that rains down to the ocean bottom, and there it gets decomposed by bacteria and then other organisms that live, crabs and other benthic, the ocean floor is called the benthic animals that feed on all those carcasses. But anyway, it's a constant recycling. The point is that of all these 2 million species, we are one of the most recent species that has developed, and we too have undergone an evolution to where we have other homins that are in our genus line, but we are the only living species as Homo sapiens. So that one was the phylogeny, in other words, the origin of the group as a whole, of the Homo genus. Then we're going to look today in this course, the beginning of life, the bioethical issues at the beginning of life, we'll look at the other one, the ontogeny. So we have phylogeny and ontogeny. Genesis being a reference to origins, right? So we're going to look at the origin of the individual. The basic question for the next 7 lectures, for these 7 lectures and modules, is when does human life begin, right? When does human life begin as such? And so the bioethical consequences, and I can say from the start to get to the punchline, is that human life begins at fertilization, all right, or conception, which is consistent with every other animal and plant that reproduces sexually. It begins at fertilization with this one single cell that is called a zygote. And that is also universal, meaning that it occurs in all species that reproduce sexually. And then the rest of the course, so that's the punchline, that's the bottom line of the course, all right? The rest of the course now will be giving the scientific evidence for that. And then also the bioethical consequences of human life beginning at fertilization. I'm sorry, let me close the door because of that. So before going any further, or any questions or comments, no? You can follow what we're going to do? Yes? All right, so we're going to look at ontogenetics, genesis meaning origin, and onto, let me start another sheet of paper here. Onto is a reference to being. All right. So I just use this as a kind of blackboard. Big enough? Right. So onto is being. And genesis is origin, just for the Latin part of the course. When we look at a thing, right, if that thing is metabolizing and is capable of reproducing, then we're looking at a living thing. And in science, we call that an organism, right? And so for an organism to be alive, it must metabolize, in other words, have body function, and must also be capable of reproducing. Those are the two fundamental characteristics of any living thing. All right. And we're focusing this first part of the course on reproduction and the two basic modes of reproduction, which are going to be asexual or sexual reproduction. Now, mechanically, what happens in reproduction is that the material of inheritance is passed on to the next generation. The inheritance material is passed on. Okay. And that inheritance material eventually was discovered to be the DNA. It's interesting to note that both Darwin and Mendel were contemporaries, but they don't not really know each other, even though, if you recall, I think I mentioned that Galton, I think he was a cousin of Darwin, showed Darwin the paper that Mendel had written about genetics, about the factors of inheritance, right, which we now call genes or chromosomes. But Darwin discarded it because he couldn't understand the math, and it was simple statistics that we were talking about. Anyway, Mendel was a mathematician. I think his father was a mathematician also. But he's the father of genetics, and Darwin the father of evolution for having come up with those theories. So we're going to be focusing on the genetics of reproduction, which is at the chemical level how inheritance occurs. And what I'm trying to say by all this is that when we look at nature, okay, when we look outside the window, there is no magic there. There's really no magic. There is a mechanical process that happens, and if all the conditions are proper, it will happen every time that way, okay? And this applies to reproduction, so that when we present arguments of credibility, we're looking at really very chemical and mechanical processes that, again, when the conditions are the same, the process repeats itself over and over, just like every time we turn the key on the car starts, because it's a mechanical process that is also tied in with electricity and so forth, but it's all matter and energy that is involved in it at the organic level. Okay, there's really magic doesn't really work. In science, it's not fictitious, in other words. All right, so what is interesting about reproduction is that this process is this single word, right, describes a process that allows the next generation to be of the same species as the previous generation. Now we take it for granted, but consider that each generation, these are individuals, these are individual organisms, okay? And how do individual organisms get to generate, really create or manufacture, is a better word, a new individual just like themselves, okay? And that's what reproduction is about. So there's a philosophical angle to this, if you will, that essentially reproduction is an elegant mechanism in nature for generating another individual essentially identical or almost identical to the previous individual. But it's another individual with its own characteristics and its own entity, its own being, all right? And these arguments are either taken for granted or really not understood by, in general, by the people who hold the legitimacy or the ethics of abortion, for example, and any other of the grave attacks that are being done at the beginning of human life today. Right? They just take it for granted, or they don't really understand that there is a natural process that determines when that individual, the new individual, begins. Okay, so we're going to look at reproduction, and let me move forward, because in nature, again, in nature, there are only two modes of reproduction. And sometimes some organisms have the capacity to use both. These are sexual and asexual reproduction. Some of this is really kind of basic, but I just want everyone to be on the same page, all right? And so we're going to establish the groundwork for the course. These two types of reproduction, sexual or asexual. Notice that the A in front of a word typically negates the word. So asexual means non-sexual reproduction. So what is sexual reproduction? Sexual reproduction involves sex. And again, when we look out the window in nature, we only find two sexes, all right? And we name them male and female, but there are basically two sexes, so it's a binary system. And again, you see that something as simple as that, and as obvious as a simple observation of the two sexes in nature, is challenged today and questioned by an increasing number of people when it comes to the human. And they get confused because they separate and make a radical separation between sex and gender. We'll get to that topic later when we talk about the healthcare bioethics. Okay, and that's it's a very dangerous and dishonest thing to do, to separate sex from gender, because it really is not that way in nature. Okay. Okay, so first I'm going to look at asexual reproduction. There are advantages and disadvantages. And I can tell you, again, getting a little to the punchline, that for asexual reproduction, for non-sexual reproduction, one of the main advantages when contrasted to sexual is that it takes less energy, less effort for the individual. Okay? In fact, it takes about half the energy if it's actually measured in kilocalories. It's estimated that sexual reproduction globally, the production of gametes and so forth, takes about twice as much energy as reproducing asexually. And that's, again, a biological, scientific fact. So it's, bottom line, it's easier for animals and plants and fungi and bacteria, okay, the four main domains, to reproduce asexually. The downside, then, the downside is that there's essentially no genetic mix, so that the progeny, our clones, are practically identical to the parents. There's no mix. So it doesn't afford for variation, you see. And remember that evolution acts on variation, right? Natural selection acts on variation, on the variance. Because certain variants may be more, better adapted to a particular climate, or especially if there's a climate change, then different variants will survive distinctly depending on how well adapted they are to the change. So the downside of asexual reproduction is precisely climate change. If there's climate change, all right, or changing habitat, then they are at risk because there's much less variation within that species. However, conversely, you know, when the climate is fairly stable, even if it's seasonal, remember that seasons are weather, not climate. Climate is long-term, long-range. But even if there's seasonal weathering, like the 4 seasons, but within the certain range of tolerance that repeats year after year, then we can say that that climate as a whole is stable. So asexual reproduction is easier because it's less energetic, right? However, sexual reproduction, which again is thought to have evolved later in species, has the advantage, even though there is more energy to investment, has the advantage that it generates variation in the progeny. And that variation, again, is mechanical, is chemical, is physical, all right? There's no magic in, for example, the puppies of a dog to be different colors, variations of the different colors of the parents, okay? There's no magic of that color variation or size variation and so forth. There's an actual explanation, and it's at 2 levels. It's going to be recombination and also the actual fertilization, the individual gametes that fertilize each other, that particular set of genes. So you, in a sense, you can see that as a bottleneck because of the millions of sperm that are produced, for example. Only one sperm is the one that's going to fertilize. So that's a huge bottleneck there. It's going to be the exact precise genes that that sperm is carrying, as opposed to the variant gene that the next sperm next to it is carrying. See? So it's a huge bottleneck there in fertilization with regards to genetics. But it does afford, the good side of this is that it affords for variation. And so it allows for natural selection to do its thing, as it were, and continuously generate species that are well adapted, generally well adapted to the climate. And that's why we see that the life process, the speciation process, is a fanning out process as opposed to a crunching down. But when we have typically catastrophic events occurring in nature, then there would be a crunch, and that's where we have these mass extinctions, which there were, I think there were 4 that we have detected so far in the geologic time scale. Okay, I'm digressing here a little bit, but just trying to point out the advantages and disadvantages of each one of these modes of reproduction. It will become more clear as we get into the actual mechanics of it and the chemistry of it. First, on asexual reproduction. Asexual reproduction has a variety of styles or mechanisms, if you will. There's a variety of mechanisms of asexual reproduction. In contrast to sexual reproduction, which is just one mechanism, basically the fusion of gametes, right? For sexual reproduction, it is fertilization. It is a fusion of gametes, male and female gametes. In some cases, it is the fusion of what is known as isogametes. In other words, there's no male or female as such. They're just gametes. In other words, the cells are haploid. We'll get to that in a minute. And it occurs mostly in echinoderms. You know what echinoderms are, anyone? There are spiny echinoderms are animals that have skin, a thin layer of skin covering their spines, their echinos. They are the starfish, the sea urchins, and those creatures, okay? They just put out what is known as isogametes. They're not male or female, but there's still a fertilization process occurring. That's kind of a specialized type of fertilization. But back to asexual, there are all these different types of, or mechanisms, if you will, of asexual reproduction. So I'm going to show you some examples, saying also that for us, as far as human life is concerned, the one that is most relevant is mitosis, right? Is mitosis. And then twinning also as a rare event in human reproduction. But mitosis, because every cell in our body essentially is undergoing mitosis on a regular basis. It's part of the cell cycle. Okay, so let's go forward here and give you some examples. Generally, they follow a pattern of more primitive to more elaborate, or more primitive organisms to more complex organisms, but that's just generally, not, it doesn't hold very strongly. So binary fission is very prevalent in bacteria and also in protozoans. Remember, protozoans was this grab bag of animals, and they're animals, but some of them are capable of photosynthesis, like the euglena. Anyway, they're typically unicellular. So unicellular organisms, the standard mode of reproduction for unicellular organisms is asexual reproduction by binary fission, okay? Fission. Now, if you remember, fusion is coming together. Fusion is coming together with one S, but fission is separating. So it's a split, essentially a split in half. You can see here individual bacteria, and this one is starting to split. This is further along. This is almost done, splitting, right? So when the cell gets essentially a little too large, the cell splits into two. And so it's a fairly straightforward mechanism, but there is one detail that is essential, which is that the DNA within these cells has to duplicate before the splitting, so that each daughter cell will end up with the same amount of DNA, okay? Keep that in the back of your mind because we'll come to that in greater detail. The thing is this, just before the cell splits into two, in whatever cell it is, the DNA has to duplicate in order for the daughter cells, the two daughter cells, to have the same amount and type of DNA, of genetic material, than the one mother cell. Okay, it makes sense, right? It makes sense. If we end up with two daughter cells that are identical to the mother cell, well, they each have to have identical DNA. But because they are two, prior to the splitting, they had to be a duplication of DNA. I'm just calling it a duplication of DNA for now. All right? Okay, I'll show you more details of that in a moment. So this is the good old amoeba. You can see that it's being timed here. Timed photography, and when it gets too large, it starts splitting in half. And finally, we have the two bacteria, which have this, the two amoeba, who are identical to the original mother cell by way of DNA and species and everything else. Budding is another type of asexual reproduction. People know what is yeast? Yeast, what do we use yeast for? Making bread, right? Okay, for rising the bread, actually rising the bread. The rising of the bread is interesting because, first of all, yeast is a type of fungus, so they're fungi. They are unicellular, but they are eukaryotes. In other words, they have a true nucleus. And they also reproduce by budding or splitting. Here you can see, for example, this one is the beginning of a bud here, further developed here, even more so here, and then finally they split and form two buds or two yeast. Which, by the way, the rising of the bread is why. When we open the bread that has been, so it's a process of what? The rising of the bread is a process of fermentation, all right? The yeast is eating up the glucose or the starch, digesting the starch in the bread, metabolizing it, and part of the metabolism is to produce CO2, which is a gas, all right, in the ambient temperature. And so that CO2 gas inside the mass of bread generates bubbles. And those bubbles are the holes inside the bread. Okay, of course, the CO2 dissipates and goes out into the environment and is innocuous, but that's basically it's a process of fermentation that makes the bread rise with bubbles of CO2. All right, this one is a Hydra. And you can see a little bud here coming out. Eventually, this bud will sever from the body of the Hydra and be a new little Hydra, which then will grow larger and generate its own buds. Hydra is a salenterate, which is in the group of, let me see, salenterates. These are jellyfish. They're not real fish, right? But they are gelatinous. Corals, anemones. Okay, they're all aquatic because they're about 99% water. It's a high content of water, and they're all jelly-like. And typically they have tentacles, which have little cells that sting. That's how they catch their prey. They're voracious feeders, and they reproduce asexually by budding. It's another example. The counterpart of that, basically in plants, is called vegetative propagation. Vegetative propagation. For example, in grass, if you pull a tuft of grass, typically you get a string of tufts that come up, right? And they're all connected by a little stem, a little horizontal stem, which is here. Okay. And so that little horizontal stem is a shoot. It shoots out, as we see here in this diagram, shoots out. And when it touches the ground again there, it will start developing a new little plant, which means that this little shoot has a lot of regeneration power. They have actually stem cells, okay? And so it generates a new plant there. And that's called vegetative propagation. It's very successful, this mode of reproduction for plants, and again, is less energetic than sexual reproduction. But you can see, you can intuit also from this particular diagram that this plant also has flowers, right? And the flower, as you know, is a sexual organ of plants. So it means that this particular plant also reproduces sexually. Okay, but it may reproduce asexually more often than sexually. And there may be years where no flowers were produced or degenerate flowers because of severe weather, but still the plant is reproducing by vegetative propagation, so it manages to generate more offspring. Then there's another example in fungi, mostly fungi, that is called sporogenesis. And it also occurs in primitive plants like mosses and ferns, sporogenesis. In other words, the spores give rise to individual plants, individual organisms, or little mushrooms, okay? And these spores typically are haploid. In other words, they don't have paired chromosomes. When they grow, then the individual plants can produce gametes and fertilize. So sporogenesis is typically in fungi, it is one half of the picture. The other half is fertilization. But the individual plant, the individual organism comes from a haploid spore, a spore that doesn't have paired chromosomes. We'll get to haploid and diploid in a little bit. I'm just giving a general picture here as another example of asexual reproduction. Another one, back to animals, is fragmentation and regeneration. It also happens in plants, by the way, for example, with grafts and things like that. But it's very prevalent now in echinoderms, which is the starfish. It's another echinoderm. You can see here, this starfish obviously lost 1 or 2 legs here. I think it actually, typically starfish have 5 legs, so it lost 1, but it's regenerating 2. So it's an anomaly, but who cares? As long as the individual gets to survive, if he has an extra arm, who cares, right? So you can see that these buds are a regeneration from a fragmentation that occurred. But even more interesting is that the fragmented leg can also grow a new starfish, all right? As you can see here, evidently this is the fragmented leg of a starfish or arm of a starfish. And it is generating the 4 other arms, okay? As long as it contains part of the central disc, In other words, if we were to draw a circle around the base of the legs here, all right, that is considered the central disc. And as long as that leg that broke off contains part of the central disc, then that will generate the other 4 legs that are missing on arms. And the reason for that, again, where are the stem cells? The stem cells are located around the central disc, all right? It also happens with lizards, for example, when a lizard loses a piece of the tail will regenerate, typically not the full length, but in contrast to this, that's not truly asexual reproduction as such, because the tail does not become a new lizard, okay? The tail is lost. But the truncated tail manages to grow a new tail, typically not the full length. It also happens in flatworms. Planaria is a free-living flatworm, can be cut up into pieces, and each piece will regenerate a new individual. So they have a lot of regeneration power, okay, and very plastic dynamic cells. The planaria, as I mentioned, is a flatworm. There are other flatworms. The group is called Platyhelminthes, just a fancy name for flatworm in Greek, okay, Platyhelminthes. One of the Platyhelminthes is Taenia, the solitary, what's it called? Parasite. Parasite. That's another name for it. Taenia solis is the tapeworm. Sorry, thank you. Tapeworm. And each segment of the tapeworm can regenerate a new tapeworm. And so unless you get the scolex, unless you get the head out of the individual, that taenia will continue to produce more segments, yeah, okay? So they have a lot of regeneration power. That's typical of parasites there. That's why they're tenacious. Okay, briefly, twinning, which we'll look at in more detail, especially from the bioethical quandary, how to get 2 individuals from 1 individual, 2 souls from 1 soul, all right? That's toward the end. But just at the biological level, twinning, monozygotic twins, is an example of natural cloning, all right? Where we have a single egg being fertilized by a single sperm, producing a single zygote, and then within the first 2 weeks of fertilization, first 2 weeks, or actually early embryonic development, that little embryo may split into two and generate two twins that are called identical twins or monozygotic, because they have essentially the same DNA. They come from a single cell, right? They come from single zygote, which was one egg fertilized by one sperm. Different from fraternal twins, which are two eggs fertilized by two sperm at the same time, and they just grow together in the womb. Okay? But those are fraternal twins, which can be boy and girl. But in identical twins, they have to be the same sex because they're fertilized by single sperm. Okay? And within 2 weeks, it can happen at the level of the zygote, the morula, the blastocyst, or even during gastrulation. So it's the first 2 weeks in the human is a window for twinning, for identical twinning to happen. Very interesting phenomenon. It's rare, but it's there, okay? So it's an example also of asexual, non-sexual reproduction after the fertilization. The fertilization itself, obviously, is sexual, but afterwards, the twinning process is asexual, right? Okay. So we have one example of asexual reproduction in the human, actually, which is with identical twins. And it's very early on in life and embryonic development. Parthenogenesis is another one. Partheno is a reference to virginity, partheno, okay? Parthenotes are virgins in Greek, and genesis, origin. So this is literally virgin birth. There are a number of reptiles and fish that have this characteristic, some lizards. Their eggs are, when the egg is mature, it is stimulated typically by a chemical process, or it could be mechanical, it could be something like temperature, but it can also be a chemical, and that egg is stimulated to start splitting, like mitosis, like regular cell division, but the egg will be haploid. In other words, the egg will be will not have the benefit of a complement DNA from the sperm. So it can happen in some lizards. Obviously, all these lizards then are females by default, okay, because there's no sperm. It happens in some snakes also, a few snakes, again, females, and in some fish, some species of sharks, it happens. You can see here they're XX, so they're all females. Okay. Parthenogenesis is rare, but it does happen, and these species are viable. They continue to live generation after generation. Finally, we get to mitosis, which is a very common process, universal. It happens essentially in all species that are alive at the cellular level. Especially multicellular organisms, the cellular division will be a split by mitosis. Okay, and here is just a slide. This is probably the skin of an onion, because I see the cells are very square. So typically, in addition to a cell membrane, it will have a cell wall. The cell wall in plants is, if you can consider a shoebox that is rigid, hard, and then inside will be the cell membrane containing the cell. It's like a balloon, an inflated balloon with water inside a shoebox, more or less. But the interesting thing is that we can see cells here, individual cells, at different stages of mitosis, of development, of splitting, all right? For example, here is a cell in interphase, not splitting, and we can see, okay, so about seeing the cell. Typically, the organelles inside the cell are translucent. In other words, they allow light to go through, all right? And so when we look at cells under the microscope, without tainting them, without staining them, we can't really see them. It just, they're translucent. All the organelles inside the cell are translucent. except the nucleus a little bit. If we play with the light source and try to diffract the light a little bit at an angle, then the chromosomes, particularly when they're condensed, will show like a little shadow, a little tiny shadow, and that's the origin of the name chromosome. Soma meaning body, chromo is a reference to color. Anyway, all this to say that these stains, in order to be able to see the nucleus and the DNA, these cells have been stained. Okay, they have been stained with a coloring that allows the nucleus to be seen. So this cell's an interface, and probably that dark dot there is the nucleolus, which is a condensation of more DNA and RNA inside the cell. You can see here the adjacent cell, the chromosomes are becoming distinct, right? So this is the beginning of the splitting of the cell, the beginning of mitosis, when the chromosomes condense further into distinct bodies. Again, that we call chromosome. Then later on, here more distinct bodies. Eventually, they line up the chromosomes while the membrane, the nuclear membrane dissolves away, and the chromosomes are now free floating inside the cell. And they line up at the equator of the cell. This is horizontal, but we call the middle of the cell the equator. All right, they line up there, and eventually the chromosomes, after they had split, as they had duplicated, one set of each chromosome will migrate to the opposite poles of the cell, opposite poles, as you can see here. So there will be a pole here and another pole, P-O-L-E, right, which pulls on the chromosomes to opposite poles of the cell. Here is further along the way, until eventually those two sets of chromosomes start coalescing into distinct nuclear bodies, and a membrane and nuclear membrane will be developed around them to engulf them. And finally, the cell will split into, here you see their condensed and opposite sides of the cell, and the cell would then split in two and generate two new cells. For example, this one, as you can see, they're small. These are the two new cells in the final process, okay? All starting from the original cell that has now split in two. And again, to reiterate, the material, the chromosome material in the one cell is identical to the chromosome material, essentially identical, of the two daughter cells. So there was a duplication of chromosomes early on, which happened at the very first stage of mitosis, which is called the prophase. Anyway, we'll look at it in a minute. So these are different modes of reproduction for asexual reproduction, different mechanisms. And you can see they're varied and they occur and they work, and they've been working for millions and millions of years. And that's how organs and tissues grow, and individuals also pass on the inheritance factors to the next generation. Staying at the cellular level, we may ask the question, well, why does this cell have to split anyway to begin with? Why does a cell have to split? Why can't it just stay as it is and just metabolize? Okay, well, the problem is a problem of surface to volume ratio, the ratio from surface to volume. Because surface is two-dimensional, right? By definition, let's say, ignoring the thickness of this paper, which is real, there is a thickness to this paper, right? Okay, but ignoring the thickness of the paper, we can say that this paper is essentially two-dimensional, right? Has a length and a width, or a length and a height, if you will, but ignoring the thickness. In contrast to the phone, for example, even though it's thin, this is three-dimensional, so this has a value. Okay, the surface of the membrane, the cellular membrane, is essentially two-dimensional. It's two-dimensional. In other words, it's a surface. It's a surface. All right. It's like the periphery of a sphere. But inside the cell is three-dimensional. There's a volume inside. And both are growing, but they grow at different rates. And this is the issue. Here's a hypothetical cell with a nucleus inside. As the cell grows, let's say that the surface duplicates, so we have twice the amount of surface. But the volume also duplicates, so we have twice the amount of volume. But the duplication of the volume is a cube operation because it's three-dimensional. So there's relatively more volume at every growth rate, at every growth stage. There's relatively more volume that is pushing against the surface of the membrane, to the point that the membrane is somewhat elastic, but at one point it's just too taut. It's like putting water into a balloon again. At some point, so the balloon will start filling, and the membrane of the balloon will stretch and stretch, but at some point, it cannot take any more volume. It will burst. And so, somehow nature has figured out that if the cell splits into two, it redistributes the ratio of volume to surface. Okay? And so by the cell splitting in two, it cuts in half the surface, but it also cuts in half the volume. So that now the two daughter cells, which start out small, again have an opportunity to stretch again and to grow more membrane and more volume until those two daughter cells get too large to hold the volume by the membrane ratio. And so they split again. Okay, so you can think of this as it's an inexorable event, for lack of another word. So it's the one that came to mind. What do I mean by something being inexorable? Okay, that it has to be that way. It's like one way. We're in a one-way street. You know the story about the one-way street, right? Yes? So this fellow was, after a party, I guess he had drunk too much, and it was going down the one-way street, but going the opposite direction. Okay? And so the cop stops him and says, "Sir, you're going on a one-way street." And he says, "Yes, officer, but I'm only going one way." He was only going one way, so it's on a one-way street. Happened to be the opposite direction. Anyway, so back to the cell splitting. What happens here is that the cell, as it metabolizes, it grows because it's accumulating more material inside, okay? And the membrane is trying to catch up by generating more and more membrane. But there comes a point that it's just too much to hold that volume into a single membrane, and so it has to split. And then when the split happens, the two daughter cells will continue to metabolize or begin metabolizing, if you will, and then accumulate more material, and again, they're stuck by the size of the volume and have to split again, okay? Then, if this is an inexorable event that is happening in our cells all the way from the zygote to the day we die, when we have billions of cells, even a baby will already have billions of cells in the body, okay? We can ask the other question, well, when does this ever stop? Because you can see that this is a geometric progression, and it accumulates numbers very fast. A geometric progression is a doubling. It's not a single linear, like 1, 2, 3, 4, 5. That's a linear progression, right? But what's geometric? 1, it's a double. So 1 and 2, yes. The linear level stops there at the second division, because after 2 will be 4. It's doubling, right? 2, 4, 8, 16. Let's say 2, 4, 8, 16, 32, 64, 128, All right, in 7 divisions, we already have 128 cells, whereas that's in a geometric progression, which is parabolic. If it were linear, in 7 divisions, we would only have 7 cells, okay, in contrast to 128. So it's a much faster accumulation of volume overall. And that's precisely what happens in the embryonic stage. It's a very fast growth of tissue and organs because of this geometric progression. Okay. But at some point, if cells don't die, then we would just become huge, too large. And gravity would take over and we wouldn't be able to move. We'd just be stuck because we have so much mass, biomass, all right? And therefore, the converse of mitosis is cell death, which is called apoptosis. And it's kind of a cellular suicide in a way, in that the cell figures out at some point that it has to die, okay? It's called apoptosis. And it's a natural cell death. Biochemically, the cell has figured out that it is too old already, and it has to die, and essentially make room for newer cells. Okay? And it's a process, what happens mechanically, and again, there's always, because there's no magic here, there's always a chemical and mechanical process that explains what happens, which is inside the cell, in addition to the nucleus, there are a number of little organelles. We'll cover this in more detail in the summer when we get into the environmental bioethics. I might as well just give you some vocabulary here. These organelles are little structures that are inside the nucleus, inside the cell, that do different particular functions, okay? For example, the nucleus. The function of the nucleus is to encase the DNA and to maintain the DNA protected. But there are many other organelles in the cell, and one of those organelles is called the lysosome, and the lysosome is like a little bubble, and inside that bubble, there are digestive enzymes that are held there until the cell has to die. And when apoptosis occurs, these lysosomes burst open, and those enzymes self-digest the cell, okay? And that's the process of natural cell death. Different cells in the body have different rates of apoptosis. Some cells last years and years and decades. Other cells are being killed or degenerating daily. For example, RBCs. Every day we replenish our RBCs, the red blood cells, to the tune of millions, whereas neurons tend to last for decades. Okay. So each cell will have its own, depending on the specialization, its own rate of apoptosis, or cell death. But anyway, this is again, you can see that it's a very mechanical thing. Why cells have to split is because of this volume to surface ratio issue. All right, now we're going to get into genetics here. Any questions about asexual reproduction? I was just giving you some examples there of asexual. Our main interest actually is sexual reproduction, but understanding asexual reproduction as a precursor to sexual reproduction will allow us to understand then fertilization in greater detail, how it is that this genetic material is passed on. Yes, Emma.
In the case of like a mother pregnancy,
Yes.
is the mitosis and everything normal from the very beginning?
Right, so molar, yes, you're talking about a hydrated form mole, right? All right, so these moles are, they can be a part of a fertilization, but it's anomalous from the very beginning, all right? And it will,
has no life.
It has a very abnormal life, and it's not really a human being because it doesn't have all the organ systems that we have. and it's not integrated as we are with all the organ systems, a dozen organ systems that work and function. So it's non-functional, and it's either discarded naturally or has to be extracted, but it's not viable, okay? TD form, well, that's whether or not it's a way. Here we go. Yeah. So this, what Henry's talking about is these molds that can form in the mother's womb. They can be the product of fertilization, but it's highly abnormal. And it's sometimes there are even teeth in here and some fragments of organs or tissues that develop, but certainly this doesn't have a harmonious integration of organs and systems to be viable outside of the womb. All right, it's just extra tissue that is being maintained alive by the circulation of the mother, but it cannot live on its own.
Even if it's a partial one?
Yes, even partial. I was trying to find one that sometimes it has bits and pieces of organs or tissues that we can recognize. So I've seen some that you can actually see parts of a cranium, parts of a head, parts of the teeth sometimes, or some hands or feet. So you can tell that it has undergone some development, but not, it's not integrated. The organs are not part of a whole system, okay? So it's definitely non-viable. It's mostly just a mass of tissue. I don't know, Doctor, you want to add anything else?
No, that's what it is.
Technically.
It's organized.
Mass of tissue, right? Yeah.
Does the woman's body think it's pregnant, like the hormone levels and everything are like maintaining it as pregnancy?
They usually don't go to full term, but in the beginning,
Right.
the body's thinking it's pregnant.
Yeah, it would develop a placenta, yes, or a link, a circulatory link to the mother. Mm-hmm. Now, some are discarded naturally, no?
Yes.
Typically, they're passed. At some point, the uterus realizes this is not a viable creature there, and is discarded. Yeah, those are hydatidiform moles. All right, so let's now go a little bit into genetics. And essentially, we're going to look at DNA, a little bit of the structure and function of DNA. All right, structure and function of DNA. Let's see where we're at here. Okay, let's see what I have self-synchron. Okay, on the handout, because this is actually conflation of two handouts. This one. Section, okay. Okay, yes, on the back side of the first handout on asexual, where it has the asexual reproduction, budding, and so forth. Okay, on the back side. I'll come back to the cell cycle in a minute. Okay, so today we know that it is DNA, the genetic material, the one that passes on the information to the progeny. The DNA is contained within the nucleus of the cell. The nucleus, by the way, is also a double membrane, just like the cellular membrane is a double membrane. And the function of that nucleus is to maintain the DNA inside that is protected. The DNA is what we call a double helix at the chemical structure, at the molecular level itself. It's a double helix, just a fancy way of saying a twisted ladder, Okay. Now, you've all seen these models of a twisted ladder. It's like taking a ladder that is long and twisting it in opposite directions. So you have two rails that intertwine, and then you have the rungs or the steps in between. And it is in the rungs or the steps that the genetic information is maintained, right inside in the steps here that are bases or base pairs. We get to base pairs. Before we go there, I just want to mention that the DNA has two ways of, two structural ways. The one way is when the cell is not reproducing, when the cell is metabolizing. So a cell is either metabolizing or reproducing, okay? Cannot do both at the same time. So when the cell is not reproducing, the cell is functioning, it's metabolizing normally, okay? Depends on where the cell is, the particular function. If it's a liver cell, it will be doing liver things. If it's a brain cell, it will be doing brain things and so forth. So all cells in the body, they're metabolizing. The metabolism is subdivided into two categories, either anabolism or catabolism, whether we're building up molecules or breaking down. Don't need to worry about that for now. But basically, metabolism is another way of saying body function, right? Metabolism. And this body function occurs at the cellular level. So there's cellular metabolism, and then the organism as a whole is metabolizing, meaning that the organism is alive. Okay, so when the cell is metabolizing, the DNA is in the form or shape of a single molecule. That single molecule is called chromatin, and it's a long molecule. It's a super long molecule. If we were to pull out the DNA molecule from a single cell, or from one of our cells, just one cell, pull out the DNA, that single molecule, you know how long it would be? It's about 6 feet, about 2 meters long, okay? Just one molecule. So you have billions and billions of atoms. It's, in fact, the base pairs are 3 billion for the human, 10 to the 9, right? That's base pairs, and there are dozens of atoms that make up those base pairs. So literally has like trillions of atoms. This one single molecule. It's possibly the largest molecule that occurs, a stable molecule that occurs in nature, if you think about it, chromatin. Contrast that, for example, with a molecule of water. What is the formula for water, the chemical formula for water? H2O. How many atoms in one molecule of water? Well, H2, so 2 hydrogens and 1 oxygen. So 3 atoms, total of 3 atoms for a molecule of water, one molecule of water, right? Okay. How about CO2? CO2, 1 carbon and 2 oxygens. So again, 3 atoms, 1 carbon and 2 oxygens, CO2. So each molecule of CO2 has 3 atoms, small, tiny, okay? How about oxygen, the oxygen that we're breathing? The oxygen that we're breathing is 2, it's diatomic oxygen, O2, right? We're also breathing nitrogen, diatomic nitrogen, N2. So many molecules are tiny, small, 1, 2, 3, 4 atoms. This molecule, single molecule, has trillions of atoms, all right? Now, how are those 6 feet put into the nucleus of a cell? Already the cell is microscopic. Now the nucleus of that cell is super microscopic. Now the chromatin inside the nucleus of that cell is super, super microscopic, okay? Well, it is put by a process called compaction. Compaction, which is the DNA winding upon itself. Compaction. It's also known as supercoiling. And it's just condensing DNA into the nucleus of the cell. All right. So compaction. It's a condensation process. In other words, the way to do this, because this molecule is huge, but it's just very long. It's long. Then it winds upon itself like a rubber band. If you take a rubber band and you start twisting it in opposite directions, it coils upon itself, all right? And it continues to coil and coil into a little wad, all right? That's the supercoiling. That's the supercoiling process. So it's a coiling process upon itself. And that single molecule then, inside the nucleus, is called chromatin. Chromatin. That's when the cell is in interphase and the cell is metabolizing. Sorry, at interphase, the cell is called, when the cell is metabolizing, it's called interphase. Interphase. In other words, the cell is not reproducing. It's between phases. It's between phases, between reproductive phases. Okay? So a cell that is not reproducing is in interphase. It is metabolizing. It is just doing its normal body function, what it's supposed to do. Now, when that cell is going to go into reproductive mode, it's going to cycle, so cycling. So it's going to cycle into reproductive mode. The DNA has to condense even further. And so you can think of this, the chromatin inside the nucleus is like having a bowl of spaghetti, but there's a single spaghetti in there. It's a single string of spaghetti, okay? That's chromatin. And it's a very long string. You pull it and keep pulling, and eventually you empty the whole bowl. Single wad of spaghetti, single string. When the cell is going to undergo reproduction, that spaghetti breaks up into distinct segments that we call chromosomes. So those are the distinct segments of the chromosomes. In the case of humans, it's going to be how many segments? It's going to be a grand total of 46 segments. But those segments are paired, okay, so that they have each segment, each two segments are the same size, the same length. So let's say we have the whole spaghetti here long. The first segment is going to be large, let's say, and the second segment is going to be the same size. Then the third segment is going to be a little smaller, and the fourth one is going to be the same size as the third, and so on down the line, so that we end up with pairs, 23 pairs. 23 pairs, and those are the chromosomes. Okay. So again, I'm talking for the human. In the chimpanzee, for example, it's 48 chromosomes, so it's 24 pairs. It happens that they have one more chromosome than we do, which doesn't make them more intelligent necessarily, because they're not, okay? But they do happen to have one more chromosome. Generally, the more chromosomes, the more sophisticated the species that it doesn't hold. There are some worms that have 200 chromosomes, okay? And they're not anywhere near as complex as we are. But we, again, back to the human number, we have 46 chromosomes or 23 pairs in every single cell of our body, except a few rare cells that have ditched out the nucleus, which are the red blood cells again, right? The mature red blood cells. Every other cell typically has in their nuclei 23 pairs of chromosomes. It is those pairs of chromosomes that need to split in order to double the number, okay, or duplicate, sorry, is the word, duplicate before the cell actually splits in two. And so we see here a chromosome that has already, first of all, it's a condensed chromosome, so it's condensed chromatin or DNA. but it has already split into two, okay, each chromosome generating a second identical twin arms, if you will. So this condensed chromosome has already split into, has duplicated, I'm sorry, the word I was looking for, has already duplicated, and that's why it's shown like this, okay? But the original segment was this one. And there's another chromosome like this one that is identical in size. And then there will be another 22 that are different in size, but each one will have its own pair at the same size. Okay, let's go further deep into the structure. Oh, before we leave this one. So yeah, the nuclear pore, because I mentioned that the membrane, the nuclear membrane is a double membrane, as you can see here. It's a double membrane. And it is essentially to hold the DNA in place and sealed and protected, right? Because that's the inheritance material right there. It's the inheritance factor. And so that cannot get damaged. If there's a mutation there, it will be passed on. The likelihood it will be passed on to the next generation. So there are these nuclear pores, which are essentially doors for communication with the cytoplasm. The DNA itself does not leave the nucleus normally. It is a messenger molecule that is sent out, which is called precisely messenger RNA. All right, so the RNA is synthesized inside the nucleus and then shipped out through the nuclear pore into the cytoplasm for generating the protein that we'll get to in a minute. But basically, the DNA remains inside the nucleus all the time. All right, let's look a little bit at the chemical structure of DNA, because this is the inheritance molecule. And as I've been saying all along, it is chemical, it is mechanical, it is physical, physiological, it functions, and there's no magic here. This is where the information is stored to be passed on to the next generation. I know I'm belaboring the point, but think of this structure, how this structure has come about, that this is also a product of evolution, the DNA itself. I don't know if you remember, but we talked about an RNA world before the DNA, which is the beginning of a complexity of a molecule that can store information. So the whole idea is the nucleic acids, as they're called, DNA and RNA, are molecules that can store information, all right? And so that's their main function. RNA is a single strand of ladder. It's like a single rail of a ladder, and it does have bases, but the bases are not paired. So you see that RNA is more delicate and prone to change, and it's prone to losing the information, because the RNA is a single rail with the bases sticking out like that, and it's unstable. So what I was looking for is unstable. And so many times the RNA hairpins upon itself to stabilize the bases. But DNA, in contrast to RNA, DNA is very stable because the bases are paired up. Okay, so these hydrogen bonds that are here are paired up, and that way the molecule is stable, chemically stable. So that's the reason for the double helix, the double helix. I mentioned that the information is stored in the bases, and this is as deep as we're going to go. We're not going to go into the actual chemical structure of the base pairs, but it's sufficient to know that the bases, what I'm calling base, is a molecule that is sticking out from the rail, all right? Like, for example, this T here, or this A over here that is upside down, okay? These are bases, and they're also known as nucleotides, and there are only 4 bases or 4 nucleotides in nature, in whatever species, naturally, all right? They each have a name: adenine, thymine, guanine, and cytosine. So these are the 4 bases in nature, the 4 DNA bases, which are also known as nucleotides. And I say this so that, this gives you competence to read scientific literature and to understand the terminology, what they're talking about. These 4 bases are abbreviated with their first letter, ATCG. And you see ATCG all over the place in scientific papers. Okay? It's just the abbreviation of the 4 letters. And they pair up. They pair up always, naturally, generally, adenine with thymine, so A with T, and guanine with cytosine, C with G. An easy way to remember that is that the straight letters pair up and the curved letters also pair up, make a match. So you see A and T are straight and C and G are curved. So the straights pair up and the curves also pair up, match up. There's a match between the straights and the curves, okay? In fact, the matching has to do with the fact that adenine and guanine are double rings. Chemically, they're double rings, whereas thymine and cytosine are single rings. So there's always a total of 3 rings in between the rails. 3 rings, okay? What does that mean? That if there were 4 rings, then the rails would kink out and not be parallel. If there were 2 rings, let's say T with C, only 2 rings, then the rails are kinking in and it's not parallel. And it makes the DNA unstable and more prone to mutation and so forth. So the fact that there are always 3 rings in between the 2 rails make the 2 rails parallel to each other. And the 3 rings are obtained by combining 2 rings and 1 ring. Adenine and guanine are the ones that have 2 rings, and thymine and cytosine have a single ring. That's why you can see here that A is twice as long as T, and G is twice as long as C. That's one aspect, but that's not the only aspect, because one can ask, well, if T and C are single rings, they essentially have the same size, right? Why cannot we pair A with C and G with T? Because the other part of the story is that at the bonding site, there are hydrogen bonds sticking out, there are hydrogens sticking out. At the bonding site, A and T only have 2 hydrogen bonding, but C and G have 3 hydrogen bonding, and that's why they couldn't combine, okay? There will be a hydrogen bond missing for C and G, or there would be a hydrogen bond lacking for A and T. So again, a little too much detail, all this just to say that this model is very stable. When you have A and T pairing up and C and G pairing up, so that the DNA molecule as a whole is extremely stable, extremely stable. It's also hydrated, meaning that it's covered with water molecules and so forth, but that's beyond the point. The fact is that the DNA molecule is very, very stable, and it also at the same time is flexible enough to wind around little balls of protein that are called histones to start the coiling process. I'll get to that in the next slide. The last thing I want to point out here is that you notice that the rails, you notice that the A here is upside down, and T and G and A and G, they're upside down, right? They're upside down for a reason. It's because the rails are parallel, but they're anti-parallel. In other words, the reading of the alphabet, if you will, is in opposite direction, all right? It's in opposite direction. So they're called anti-parallel. Actually, I have a slide about that here. Yeah. Yeah, so the DNA molecule, the rails are anti-parallel, means that the reading of the alphabet, of the information, is going to be complementary, but going in opposite direction, in opposite read. It's like reading a book, from left to right or from right to left. One last thing about this slide here of the ATCG, of the 4 base pairs. all DNA normally, these are the 4 base pairs. So the alphabet only contains 4 letters. And with 4 letters, it is how they are sequenced, how they are next to each other, the particular sequence that determines the gene, determines the information, right? So with only 4 letters, distributing them in a particular sequence will generate the genes that are necessary for the proteins that form all the body. By the way, another detail is that these sequences of bases, they code for making proteins. So what DNA does at the first level of interaction, at the first level is the DNA makes proteins. Then proteins do the work in the body. Proteins then will metabolize, okay, and do the work in the body. It is the proteins that do the work. The DNA, the work, if you will, or the task of the DNA is to generate proteins by a particular sequence. So by changing the sequence, the DNA can generate literally thousands and hundreds of thousands of different proteins that do the actual work, the specific work on the cells of the body, the tissue that we're talking about. All right. Let's go back into the cell or into the nucleus with the DNA, starting with the double helix, right? The twisted ladder and the base pairing, that's stable. So that double helix will also wind up, because it is so long, it will wind up around a little ball of proteins. They're called histones. I have it here somewhere. Yeah, it's a little faint. Oh yeah, here it goes. And yeah, there are 8. There are four pairs of histones. There's histone 2A, 2B, 3A, 3B, 4A, 4B. They form a little ball. And this ball is like the center of a yo-yo, where the string winds around the yo-yo. Okay, it's the core. And so the DNA winds around the histone proteins almost 2 times, 1.7 times. Don't have to worry about that detail, but it's almost 2 times 1. And that complex of protein and DNA is called the nucleosome, And the nucleosome is what we call the functional unit of DNA, functional unit. So you can intuit that for each nucleosome, the DNA is super stable. It's stable first because the bases are paired, and because the ladder is a twisted ladder that the rails are parallel or perfectly parallel. But in addition to that, that stable molecule is also winding around these little protein balls that further stabilizes the molecule, and that begins the compaction process. All right? It's winding around the DNA, these histone balls. Then there will be a little segment of DNA to the next histone ball, and we get an arrangement at this level. We get an arrangement that is known as beads on a string. Beads on a string. It almost looks like pearls on a collar, right? That you have these little balls on a string. So it's called beads on a string. But that is not sufficient to put in 6 feet of molecule inside a nucleus. It has to wind again upon itself further. All right. So the little link DNA between nucleosomes is also flexible enough to allow more winding. So here's where we get into the supercoiling. First, there's the coiling around the nucleosome. Then there's a supercoiling of nucleosome upon nucleosome upon nucleosome. It's a very geometric arrangement. And it continues to wind around. So it's like threading a rope, if you will, thicker and thicker. Then that stabilizes, but it's still not enough. It will continue to coil again upon itself, and another layer, until it's just totally compacted to the level that it becomes a chromosome that can now fit into the nucleus. And the level of compaction is, how much do you think is needed? For example, the folding, it's like a folding upon itself, right? So if I fold this paper into half, so that's one level of folding, right? Another level of folding is 2 levels of folding. How many sheets of paper do I have? 4, okay? Now it's the same surface, but it's being compacted into a smaller size, right? Now fold it again, it's the 3rd level. Now there are 8 layers. 8 layers, 4th level, 16 layers, 5th level, 32 layers, 6th level, 64 layers. Anyone want to try to fold that again?
It's too hard.
Exactly. So we went 8 levels, right? And we got 64 folding over. The DNA molecule to compact into the nucleus compacts about 10,000 fold. 10,000 fold. And that's how you get 6 feet into the microscopic nucleus of a microscopic cell. 10,000 fold of the DNA winding upon itself. So it's super compacted. That means that it is incredibly inaccessible. It's definitely, that information is so tight in there, is no one's going to get, no molecules are going to get in there to read that DNA out. It's super compacted. That's because the cell is going into reproductive mode, and it's going to stop the metabolism, except for the metabolism that is associated with reproduction itself, okay? Now, before it does the compaction, the full compaction, the chromatin will duplicate. And so when you see chromosomes designed like this, all right, or illustrated like this, and you see kind of an X-shaped form, this tells you that this chromosome has already duplicated, but is being held together in the middle by just a structure that is called the centromere. But when you see chromosomes represented in an X shape, they have already duplicated, all right? So this arm is a duplicate here, and this arm or leg is a duplicate here. But they're being held together as a single unit. So there would be, how many of these? 23 pairs, 23 pairs, or 46 of this, because the duplication has already occurred, but they're held together as a distinct chromosome. All right, so we cover that. We cover the antiparallel. Let's look at this duplication process briefly, then I think we can break after this, just to complete this story of the duplication. Right, when we see the chromosome represented like this, duplication has already occurred, but they're still held together by a central structure there called the centrosome. All right. The duplication machinery, or the duplication process, because we scientists, we don't want people to understand us all the way, all the time, so we use sophisticated words. It's called replication. So duplication in science is called replication, all right? So just substitute the du for the re. And replication means doubling up the chromosome or the DNA material. And this is the replication machinery, which involves several different proteins. See here, there is this thing called a helicase. The helicase is splitting the pairs, splitting the bases right down the middle, separating the pairs. Separated, so it basically is breaking hydrogen bonds, okay, and separating the pairs. And then the replication machinery proper comes in, which are known as polymerases. Polymerases, this yellow structure here is actually a complex of proteins put together. And what they do is they attach nucleotides that are floating in the cytoplasm. There's a supply, if you will, of nucleotides in the cytoplasm, and they're being attached together, bonded together by this polymerase molecule. Essentially, they are rebonding, reestablishing the hydrogen bonds. Accordingly, the A's with the T's and the G's with the C's and vice versa and so forth to regenerate the double helix and to stabilize the molecule again, all right? It happens on the what is called the leading strand, and it also happens on the lagging strand. The lagging strand has to be done by segments because, well, there's a chemical process here. The machinery always goes 5' to 3', but that's aside the point. The fact is that when the DNA is split into half, each half is duplicated with its complement. So whatever was a T alone will hook up an A. And if there was a G alone, a C would come in and do the complement. So they complement each other. The two strands complement each other. All right? What else to point out? So here is the duplication is occurring, which we call replication. And the end result is precisely this chromosome with its double legs. Oh, I mentioned earlier that DNA has the information for proteins, but it is the actual proteins that do the work. And here we have the case in point. We see precisely the helicase is a protein. The polymerases, they're also proteins. Typically, when you see a word that ends in ASE, it's a protein, it's an enzyme. So all enzymes are proteins. Not all proteins are enzymes. There are other proteins that are structural proteins, etc. But all enzymes are proteins, and they do the work, the actual physical work that needs to happen in the cell for metabolism. Okay? So the duplication of chromosomes, replication. And it doubles the genetic material, but it's complementary. So this double helix over here is going to be complementary to this other one here, okay? Also, I mentioned that the DNA stays within the nucleus, and what comes out is a message of the sequence, of the particular gene sequence that is needed, the particular protein that is needed for that function of that cell. And so the message is delivered out of the nucleus through another protein, another nucleic acid that is called now RNA, ribonucleic acid, RNA, and this is messenger RNA. So little m in front of it represents messenger RNA. Okay? And it's the messenger RNA that comes out through the nuclear pores into the cytoplasm. In the cytoplasm, what we have is other proteins, protein complex, that read the messenger RNA, and by reading the messenger RNA, the particular sequence of bases, it will match up amino acids to those bases. And so just like nucleotides are the building blocks of DNA, amino acids are the building blocks of proteins. Okay? Think of it this way. Ever get stuck on the train here in Hialeah when you're going up and down? What do? We sit there, we start counting cars, right? Okay, so they're like cards. The cards of the wagons of a train are like the amino acids of a protein. They're the basic building blocks of a protein are these amino acids, all right? Now, they're amino acids of different types. It happens that in nature, there are 20 different types of amino acids. Just like there are 4 bases in nature, 4 nucleotides, right? There are 20 amino acids. I'm not asking you to memorize the amino acids, just know that there's a larger number of amino acids. There are 20 of them. Hmm? And it's like that train again in Hialeah, first, let's say, has 3 locomotives. Okay, well, 3 locomotives is going to be a long train with 3 locomotives. Those 3 locomotives represent one particular amino acid, let's say alanine. Then come 5 wagons of concrete blocks. 5 wagons of concrete blocks, so that's going to be lysine. All right, so you got 5 amino acid lysine. Then come 20 wagons of containers, the shipping containers. Okay, so that's tyrosine, it's another amino acid, and so forth. All these amino acids will be sequenced like that in a single string. And that's what we see here, okay? So in addition to replication, which happens where? Where in the cell does replication happen? When I say replication, you think duplication. What's duplicating? The nucleus, right? The DNA. So replication happens in the nucleus. It's a duplication of DNA in the nucleus. In addition to that function, the DNA also sends out this information to the cytoplasm. And that's not replication, that's going to be transcription. And so that process is known as the central dogma, transcription and translation. It's a two-step thing. The first step occurs inside the nucleus when DNA generates an RNA molecule, an RNA, which is a single strand. Okay? The DNA is being transcribed into an RNA. It's like a photocopy. So you have the DNA, which is the original. You take a photocopy of that original. That's the RNA. That process is called transcription. It's a transcribing process. The information is passed to another nucleic acid, to the RNA. That RNA is shipped out of the nucleus, goes to the cytoplasm, to particular proteins called ribosomes. These are also organelles. These are organelles in the cell. And the ribosomes then translate the RNA into protein. And that's the second part of the process, the translation. So there's transcription and translation. Transcription is going from DNA to RNA. Where does that occur? In the nucleus. And then translation is going from RNA to protein, and that occurs in the cytoplasm. All right? So it's a two-step process. So what do we see in transcription? From DNA to RNA is passing the information, you see? So that way the DNA does not have to leave the nucleus because it's dangerous out there. And if there's a mutation, then that it's going to pass on, and that's going to damage the cell. So the DNA transcribes, the original never leaves the museum. We make a copy, and the copy will leave. That's the RNA. Also, at the level of detail, RNA does not have thymine. Instead of thymine, it has uracil. It's a detail, but it's important because the DNA code, the genetic code is given in RNA terms, not DNA. And you'll see U instead of T. So just don't worry when you see U for a genetic code. It's just that they're giving you the RNA version, which U is a substitute for T. All right. And then the full name is just uracil instead of thymine, and that's in the RNA. So RNA has uracil instead of thymine. That's one change. The other change is that it's a single rail instead of a double rail, and that's about it. All right. The information is stored in triplets. Just know that it's a triplet code. In other words, it's 3 nucleotides in a sequence, 3 bases in a sequence, okay? It's a triplet code. So let's say that the top, this is the original DNA here, and the strand that is going to be transcribed is the top one, the ATGA, right? That's called the coding strand because that contains the code, the genetic code. That is transcribed into the RNA that you see here in red or pink, all right? And so you see that the RNA is equivalent to the coding strand. To be equivalent to the coding strand, it was pulled out of the template strand because the template is a complementary. All right, so the template is a complementary. For example, just concentrate on the first 3 nucleotides here. What are the first 3 nucleotides on the coding strand? ATG, right? ATG. We want that message to go out. We want the ATG protein, if you will, or the ATG amino acid to be translated in the ribosome. So we're going to get that sequence of RNA by using the template strand, by using the complementary strand, right? In other words, what we need for the RNA to have, what is the RNA equivalent of ATG? The RNA equivalent of ATG is AUG, right? Because thymine is replaced by uracil, right? So that's the RNA equivalent. See, we want this is the code is here, all right? And we want this amino acid, ATG amino acid. So it uses the transcription machinery is going to use the template strand, which has the complement. In other words, TAC. We need it backwards. We need the complement. TAC, in order for the RNA to be AUG and match up to the coding strand. Okay? Okay, so now we have, that's why it's called the transcript of the coding strand. This RNA is the one that's shipped out through the nuclear pore, and now we go into the translation segment, which is from RNA to proteins, the big picture. What we have actually is that each one of this triplet is going to code for a particular amino acid. So AUG is going to code for methionine, AUC is going to code for isoleucine, and AUC and UCG is going to code for serine, all right, which are 3 amino acids. When amino acids are linked together, the intermediate molecule is called a polypeptide. It's the intermediate molecule. So a series of amino acids forms a polypeptide, and then polypeptides coming together into longer strands form the actual protein. So the polypeptides are like an intermediate structure between the amino acid, the unit, the single unit, and the actual functional protein. Okay, now let's shift to the protein for a moment, just for the sake of completion, so that you have the full picture here. Again, maybe it's a little bit too much detail, but we have another molecule that is informative in the sense that it's a chain of amino acids linked together. But in this case, it's a single chain of amino acids. It's not as complicated as the DNA structure with a rail and the rungs and so forth. Just amino acids linked serially, right, in series, but they do wind upon each other for stability. And they wind and they form either alpha helices or beta sheets, which are the secondary, it's known as the secondary structure of proteins. These 2 basic structures, alpha helices and beta sheets, these also are already 3-dimensional. And when they come together, these subunits come together, they form what is known as a tertiary structure. And these are the subunits of the protein complex as such. Give you an example. This final protein complex, which is known as the quaternary structure, right, because it has undergone 4 layers of complexity or 4 layers of folding. The first layer is the amino acid sequence. The second layer is the alpha helix or beta sheets. The third level is the subunits coming together. And finally, those subunits making the functional protein. This one example is hemoglobin, which is the one that carries oxygen inside the RBC. The hemoglobin is a protein complex that is made up of 4 subunits. Each subunit is a myoglobin protein, okay? And 4 myoglobins coming together form the functional hemoglobin structure. So you get complexity upon complexity. And it is the hemoglobin protein complex that is functional as a whole for metabolism. In other words, the myoglobin, each one of these 4 myoglobin subunits by itself is not capable of carrying the oxygen, all right? And so on, further back to the lower levels of complexity, you have to have the quaternary complex in order for it to be actually functional, because it has to do where the functional groups are, the particular three-dimensional phase that is being exposed to the rest of the cytoplasm for the protein to function properly. So just like DNA winds upon itself, also proteins wind upon themselves to form what we call complexes. And those complexes are the ones that are functional. For example, when I talked about the polymerase, the replication, for example, here, this is a complex. This is a DNA polymerase complex. So there are several proteins involved in making this complex for it to be functional and do its work. So that's really how enzymes work at this level of complexity, at the quaternary structural level. This is a whole thing put together now as a single map, if you will, and it's known as the central dogma in molecular biology. Just like the central dogma for population biology is evolution, the central dogma for molecular biology is from DNA to RNA, from RNA to protein. This explains, in a very simplistic way, if you will, it explains the possibility of life, of organic life, at the cellular level. And when we say cellular, we also mean tissue and organ and organism, all right? So at the cellular level, at the molecular level, this central dogma, DNA to RNA to protein, is, as you can see, it's a two-step thing. From DNA to RNA is transcription. From RNA to protein is translation. It's a little diagram of it. And like I said, this central dogma is the molecular equivalent of evolution at the population level. Okay, so we've gone from the largest possibility in nature to the smallest possibility in nature to have something living, functioning, organically speaking. Okay, why don't we stop here and break? Because it's over 11 now, going for almost 2 hours. So take a break to thaw out a little bit and come back about 15 minutes more or less. All right, so I'm going to pause this, pause recording. Okay, welcome back everyone. Let's dim the lights here. All right, and we're back to the second part of this first lecture. Again, this is like a summary of the central dogma in molecular biology that holds. When we say central dogma, it means that this is the universal level. In other words, this is how DNA and RNA and proteins behave in all species, from the simplest bacteria that we can consider all the way up to the most complex living organism today, which is the human. And it really does apply universally. DNA is the molecule of inheritance today. It may have been RNA previously, but RNA is unstable and it tends to hairpin to stabilize itself, and that's the precursor of a DNA molecule. And when we say that this applies to every species across the board, we say that this is an elegant model. Nature is known for being elegant that way. That's how we use the phrase in nature or in science, when something is so, it functions so well that it's applicable to all living organisms. Okay, there's no other mechanism of passing on the genetic information, the inheritance, than this one, because this one works. So nature is also economic. In other words, if something works, don't fix it, keep using it because it works. Having said that, I also want to point out a few more details, just because I think it's interesting. We'll go back to the replication machinery, for example, over here. Okay. This happens in fractions of seconds. It's extremely fast. And that's, enzymes are that way, in fractions of seconds. And therefore, we can say that this machinery is accurate to 99.999% of the time. Okay, you can take it down to millesimal, if you will, decimal, centesimal, millesimal. But it still leaves the 0.0001% possibility of error. And it does happen. So this machinery is not 100%, 100.000% accurate, all right? There's a little bit of error here. It means, what does that mean? That every now and then the wrong base would be put in there. Mm-hmm. That is one type of mutation. But also, I want to say that this machinery that happens in fractions of seconds, there's another machinery that comes afterwards, reading the DNA and proofreading the DNA. And when it finds an error, it corrects that error. So it takes it down to, it takes it up to another order of magnitude of accuracy. But it's still, it will never reach a full 100%. Okay, so that's another very tiny source of variation, if you will. But when we're talking about 3 billion base pairs, 10 to the 9, all right? It's a 3 and then 9 zeros. You can see that even a 99.99999% accuracy still leaves a significant number of nucleotides that could be in error. Statistically, I'm shifting into a probabilistic model here, but just to let you know that even if this happens, there's a trade-off. Making it fast, because you can see that this molecule here, when it's unzipped, it's exposed and it's prone to mutations and kinks and damage to the genetic, to the original, because this is the chromosome, this is the original, all right? So that's why this replication machinery moves within seconds to do it very fast and to try to have the DNA open for a minimal, absolutely minimal amount of time to be replicated. The downside of that is that because it's done so fast, it's prone to errors. Like when we're typing, if we touch type and we start typing too fast, we're going to be making errors, and we'll have to go back and proofread and correct that. And even if we proofread the first time, the second time we go through, we're still going to find a few more, and so on and so forth. So it's a proofreading mechanism that goes through afterwards. Imagine, and all that has evolved through evolution, again, with no magic. No one has placed it there in particular. Rather, the most that we could say from our faith is that God created and generated the fundamental principles for this to occur, but did not, for example, God did not create the DNA polymerase directly, or this DNA molecule directly, you know, like saying, let it be, boom, there it is. No, it came through an evolutionary process of billions and billions of years, beginning with elemental atoms from the universe coalescing into the earth and so forth. Okay, so those basic fundamental principles may have been established by God, but then the rest of it is just a consequence, a playing out of these principles that he set up. Plus providing the original material, right, which we call it the Big Bang in colloquial terms, because you have the principles, but you don't have an actual application where to apply those principles, then nothing happens. Mm-hmm. All right, so many details are involved in this, but imagine the complexity of these machineries to do this and to do it automatically whenever the occasion arises that the cell knows it needs to replicate, and also it does that. Okay. So the summary then is this, that on one hand, the DNA really has kind of two functions. One function is to, if you will, metabolize the metabolism of the DNA, or the function of the DNA is to produce proteins, and it will do it through, by the application of the central dogma. In other words, originally really just transcribing. In fact, after the DNA is transcribed, as far as that metabolic process is concerned, the DNA is done, because then it's the RNA's responsibility to translate into a protein. So even in the central dogma, the DNA only plays a part in the first half, in the transcription, okay? And the second part is played by the RNA, when the RNA then is translated into protein, but that happens in the cytoplasm. The other function of DNA, when it's not metabolizing, in other words, when it's not generating proteins, right? Which again do the work. The other function is to replicate, to duplicate, when the cell as a whole is going to go into duplication mode. And there, what we have is from one DNA strand, we're going to end up with 2 DNA strands. So the arithmetic really is really simple. It's duplicating, all right? It's duplicating. That duplication is done by the replication machinery, and there are complex, protein complexes that are in charge of that. And that replication process is called semi-conservative. Semi-conservative. It's another technical term in genetics. Semi-conservative means this. Let's go back here for a moment. Here we have transcription, right? Remember, there's a coding strand of DNA and then the template strand of DNA. The template strand is the complement of the coding, and the coding, by definition, is a complement of the template, right? Just like male is a complement of female, female is a complement of male, right? So it's also a binary system. It's called semi-conservative because only one of the 2 strands is being transcribed. Only one of the 2 strands is being transcribed, okay? And by doing that, it is the complementary strand that is conserved. So the RNA only has one of the strands. That's for transcription. But it also happens in replication. Think about this. We have an original strand of DNA that is stable, meaning it's base paired, coiled, all right? Now it's going to replicate, it's going to duplicate. The pairing is split up down the middle so that the bases become individual bases, and then they are complemented with their complementary nucleotide. But you see, each new strand, so from one strand we get 2 strands. Each one of the 2 strands, the 2 resultant strands, has one of the strands is the original. So let's say in this one that is, the diagram is completed, okay? Here's the original DNA split in half, then each half is complemented with a space pairing to make it a double strand. So each one of the resulting strands has one strand of the original, and the other strand was synthesized, was new. And that's what it means semi-conservative, because of the 2 resulting strands, the 2 resulting double helices, one of those helices is the original helix. It's the original helix. It's the original stair, if you will. Only one side, one of the rails. Another way of saying it, of the original double rails, one rail goes to the new strand and the other rail goes to the other new strand. So each new strand has one of the original rails. And that's what it means semi-conservative. It conserves half of the original molecule. Each resulting molecule conserves half of the original. So that's guaranteeing the accuracy, the fidelity, sorry, it guarantees the fidelity of the message. Because each second ladder has one of the rails is from the original ladder. And that is the guarantee of the fidelity of the message. And they are complementary strands. So that, again, just probably giving you a little too much detail here really, but to explain the words, if ever you see them, again, semi-conservative replication is semi-conservative, meaning that of the 2 resulting double helices, one of those helices in each strand is from the original one. And then that process continues on and on and on and on every time the cell reproduces. Okay. To finish the argument here, this now is based on translation. This is the genetic code. This is what Watson and Crick deciphered finally in the 60s. First in the 50s, 53, they deciphered the structure, the chemical structure of DNA. And it took almost another 10 years to decipher the actual code of the DNA. In other words, the fact that it's in triplets, the code is in triplets, and what each triplet codes for, what amino acid it codes for, right? So you notice that all the letters in here are triplets, right? They're all triplets. And that's what is called a codon. So a codon in DNA, in the DNA sequence, is a triplet that codes for a particular amino acid. I also mentioned, I think earlier, that the vast majority of DNA is called junk DNA. It doesn't code for any particular protein. So if we look at only the 3% fraction of those 2 meters, right? What's 3% of 2 meters? Well, let's say it's 1 meter for the sake of argument, and then we can double the number. What's 3% of 1 meter? It's 100 centimeters. It's 100 centimeters in 1 meter, right? So it'll be 3 centimeters, it's 3%. 3 centimeters is 3% of 1 meter. I have it right here. Okay, so see the little marks, not the little ticks, but the big ticks that have a number, those are centimeters. So this is 100 centimeters here. So it means 100 of them. So 3% is 3 of these. All right, 3 centimeters. But it's actually 2 meters, so we talk about 6 centimeters. 6 centimeters of the 2 meters is the coding region, very small. It will be this plus this other thing over here. Only this little part is coding for the proteins, for all of the proteins, the thousands and thousands of proteins that make up our body, of the whole DNA strand. This rest 97%, we call junk, in that there is no coding region here. Now, that's a big to-do, right? That's a big feat, how to figure out of all these little triplets, which there are 3 billion of them lined up, all right? 3 billion of these little nucleotides, how to figure out the triplets that code, all right? That was a fascinating thing to do in the 60s before the computer. Now, since then, it has been corroborated. It took a lot of experiments, but they got it. And this is the triplet code. What you see on the letters are the codons. And what you see in little abbreviations are the 20 amino acids. So if you count these separate, the separate ones, for example, this is serine, this is proline, tyrosine, alanine, aspartame, glutamine or glutamic acid, lysine, etc., arginine, serine, all right? You will count them, you will find 20. Now, how does the triplet code work? Well, we need 3 sides of the square to come for 3. So you start here, for example, and this is the first letter on the triplet. This is the first letter. This one is going to be the second letter on the triplet, and this one is going to be the third letter. We only need 3 sides to have the 3 triplets. So let's take at the very first one here. You see, this block, the first letter is U, the second letter is U, and the third letter is also U. Now the second one, the first letter is U, the second letter is also U, but the third letter can also be C. The third one, UUA, we got U, and then we got A over here, okay? And then finally the fourth one, UUG, we have the U, and then G, right? So that's how we come up with these triplets. You notice that, for example, phenylalanine, represented as PHE, is one of the 20 amino acids. 2 triplets code for phenylalanine. 2 triplets. UUU or UUC. First of all, you notice that there's a U, there's no T in here, right? Which means that this is not, truly, this is not the DNA code. Strictly speaking, which code is this? This is the RNA code. In other words, this is already at translation level, okay? But of course, if we want the DNA equivalent, what's the DNA equivalent of this first triplet? TTT, the first codon, the DNA template in the coding region is going to be TTT. All right, so this is going through transcription and translation. The translation is that codes for phenylalanine. All right, but also UUC codes for phenylalanine. For leucine, for example, LEU, UUA, or UUG, and so on down the line. You notice that at the end, what wobbles is the third triplet. And that's precisely what it's called. It's called wobbly. In other words, the third triplet is interchangeable. In some cases, you can use two different nucleotides, like in these two cases, phenylalanine and leucine. U or C works for the same amino acid. In some cases, you can use all four on the third base. For example, here for leucine, right? You can have U, C, A, or G on the third triplet, the third nucleotide. That's called wobbling. And so there are more triplets, if you will, than there are amino acids. And that's what is called redundancy, because there are 64 triplets. When we do the math and look at all the possible combinations of 4 letters in 3 packages, in 3 sets, we come up to 64, because it's 4 to the cube. Okay? In other words, what are all the possible combinations of 3 letters, of 4 letters in 3 packages, in sets of 3? It's 4 times 4 times 4, and that's 64, okay? So we have 64 triplets, 64 codons, but there are only 20 amino acids. It means that more than one codon, more than one different codon, codes for the same amino acid. And that is called redundancy. So the DNA code is redundant. There are 64 possible triplet combinations, 64 different codons, but only 20 amino acids. And that means that several different codons can code for the same amino acid. And that's the example, okay? Valine, for example, all 4. In fact, leucine has 6, because it has these 2 and these 4 also, which are different, and so forth. There's also one codon that doesn't code for any amino acids, and that's the start codon, which is AUG. Should be in here somewhere. No. But it has methylaniline. Okay, well, anyway, this always starts a coding sequence. AUG always starts a coding sequence. So there's a methylaniline amino acid always at the beginning of every polypeptide. And then there are 3 that are known as stop codons. Stop codons means that it's a triplet sequence, but it doesn't code for any amino acid. Okay, so that one, then the protein falls off. There's no more wagon to attach to the train. So that train falls off, and that completes one polypeptide. And these are the 3 stop codons here. UAA with a wobble, UAG, and then UGA also. These 3 are stop codons, meaning that in the translation machinery, in the ribosome, when the ribosome gets to any one of these 3 codons on the RNA, that polypeptide falls off because there's no other wagon to attach.
It doesn't codify to any amino acid.
Exactly.
It's like a tail.
It is a tail, right? It's a tail. It's called a stop codon. It's like it doesn't have a hook to latch on to the next amino acid. All right. This is all at the chemical level, but yeah, and there are 3 of them. There are 3 stop codons.
And every polypeptide has it, or?
Yeah. So the end of every polypeptide is ended precisely because after that one, there was after the last amino acid, there was a stop codon. And there was no matching amino acid, so the chain was broken. The chain was stopped. If you think about this functionally, that's one of the dangers of mutations, is that it will change. Let's say that you have this codon, which is tyrosine, TYR, all right? You have UAU or UAC, so it's got a wobble of 2. But if that T or that C gets mutated in the DNA to an A or a G, it will turn into a stop codon. And it will cut that polypeptide prematurely. And it will be a short polypeptide, and that polypeptide is truncated prematurely, and it may be truncated before the active site of the protein. So that protein lost the active site of its enzymatic capacity, so that's going to be a mutation, that's going to be a malfunctioning protein. It could be a vital protein in the metabolism of the body, or it could be a secondary thing. For example, if it goes for insulin and it's missing the active site of insulin, that's going to cause diabetes, which can have its consequences. So that's why mutations are dangerous. Most mutations are not dangerous because on the 3 billion base pairs, first of all, 97% of it can be mutated all you want, and there's no coding there. But in that 3%, that's where the danger is. Yeah, and that's why on average, most mutations are either neutral or sometimes they may be beneficial because they introduce something that is positive. But the few that are negative can be very damaging indeed and can lead to, typically, it alters the reproductive cycle so that the cell does not know when to stop reproducing, and the end result of that is cancerous tumors. Emma?
Does this also work together with knowing the body's enzymes, like the cardiac enzymes, pancreatic enzymes, and the liver enzymes?
Yeah, absolutely, because all those enzymes that you mentioned are specific. In other words, the cardiac enzymes will not work in the liver, and the liver enzymes will not work in the pancreas, and so forth. They're specific, they're tissue specific. Right, they're tissue specific, all right? So that brings up another point too that I'll mention as soon as Dr. Claire comes back, but which has to do with the fact that the DNA, most of the DNA is shut down most of the time because each cell contains the entire genome. Wow. Each cell contains the entire genome. And that's why we can clone a new individual from a single cell of another individual, because that cell, in principle, contains the entire genome. Okay, so this is basically, yep.
But I didn't get the 3%. I mean, this 3% is the one that is important for mutations. I mean,
yes, because that's the coding region, right? In other words, the 3% is the region that codes. What do we mean by coding region? It's the one that actually has codons, that has functional triplets that could code for a protein or for a polypeptide. Exactly, it's nonsense. We call it nonsense. There are no actual, so in other words, when you count these, all right, these come up to the 64 DNA triplets, but other combinations don't code for any particular protein sequence. And that's called nonsense sequence, because it doesn't make sense. It doesn't make coding sense.
Maybe it has a function that we...
Exactly. And that's why calling it junk, we always put it in quotations, junk, okay? Because it's junk as far as protein coding is concerned. Exactly. And that's part of my PhD research was on that at Purdue, is the structure and the relationship between structure and function of DNA. It may have what we call a higher order function. Higher order function is precisely in the winding, in the supercoiling of the DNA. It may have a function there in that some of these regions are more flexible than others, some are more rigid and so forth, so it may have a function in the way the DNA wraps around itself in supercoiling to make the final structure, all right? And so it may have regions that are more open to transcription than others, and so it may have a function there, but that's still being explored.
But every chromosome has a 3%, right?
Yes, so every chromosome has genes in it, and the genes are the coding region. You have to think in terms of, because of your biology background, think in terms of introns and exons. The exons are the ones that code, and the introns, which are in between, don't code. All right. So it's not that, you know, the 6 foot molecule, only the first, or let's go in centimeters of the 2 meter molecule, only the first 6 centimeters. That all the coding is there. No, that coding is interspersed throughout the whole molecule. It's interspersed. Sorry?
It's a 3%.
It's a 3%, exactly. But it's not the tip. All right.
Okay.
It's interspersed throughout the whole region. And then the other 97% is also interspersed in between. But there are wide spaces between genes, basically.
Distributed in the way that every
Distributed
chromosome
is the word.
has
Exactly, it's distributed, and it's not distributed homogeneously either. It's a heterogeneous distribution, which is typical of nature, is clusters. When you look at trees, tree canopies from a balloon or from a helicopter down or from Google Map, all right, you see clusters. The typical thing in nature is clusters. You don't see trees lined up in a rows, you know, homogeneously distributed. It's very seldom that you have homogeneity. Depends on what level in nature, but the typical thing in nature is like clusters. The same with genes. Genes cluster to form gene clusters, all right? And typically those genes would be related to a particular function in synchrony, that all those proteins need to be expressed more or less simultaneously to have the full function of the metabolism that is necessary. So we have clusters in the genome, gene clusters. All right, what I wanted to mention then, it could be related to this. This is just to show the distinction between DNA and RNA at the chemical level, and three basic distinctions. The most obvious one is that in contrast to DNA, RNA is a single strand, yes, a single ladder, all right, single rail, as opposed to DNA, which means that the bases are in principle exposed, they're not paired, they're not complemented, and so that makes for biochemical instability, because this A, let's say that this blue one is, blue is A, all right? So we got A, C, G, A again. So these bases will tend to pair up with these bases down here, complementary, all right? They will tend to kink around like that, and it's called hair pinning, like a hairpin from the woman, all right, what they use. The RNA will tend to fold upon itself and complement in bits and pieces, because it's not going to be totally sequential, because it's not, again, the distribution is not homogeneous, but it will hairpin in different parts and form loops and bubbles and so forth, which, because that molecule is functionally tends to stabilize itself. And that could have been the precursor for eventually the DNA molecule. But as it is, RNA is single-stranded, which means that this area also needs to be coded to maintain it that way, at least until it gets to the cytoplasm, until it gets to the ribosome, for it to be translated into the proper protein. So that's one difference. The other difference is that there is uracil instead of thymine. You can see that there are single rings as opposed to double rings, right? So they're complementary that way. And the chemical structure is slightly different. It still has oxygen here, hydrogen, oxygen, but here this radical is different. This has HC, and this is the H. So it's a little different on this side, and that makes a difference between thymin and uracil. Just the way it is. It started early and it stuck, so that's the RNA molecule. And the other one is that it has ribose instead of deoxyribose. So the ribose ring, which is part of the rail itself, the ribose is what connects phosphate to phosphates to form the rail. It's a single ribose instead of being a deoxyribose. But the full extent of the word, a lot of people hear about DNA and RNA, but they don't know the full extent of the word. Okay, so what the word means spelled out is deoxyribonucleic acid. And both are these nucleic acids. Nucleic acids is one of the 4 basic macromolecules in nature, okay, nucleic acids. The other 3 are carbohydrates, proteins, and lipids. We have proteins, lipids, carbohydrates, and nucleic acids. Nucleic acids are only these 2, DNA and RNA. Lipids are many, carbohydrates are many, including sugars, starches, etc. And proteins, again, are many. But the ones that it's a small group, the exclusive group, if you will, are the nucleic acids, which are DNA and RNA. The other thing I wanted to mention was with regards to the compaction issue. Each cell in the body has the full genome, the full 3 billion base pairs. So each cell has all of the genes in the body, whether it's a liver cell, a toe cell, a brain cell, whatever cell you think of, okay, of the 220, more or less 200 cell lines that we have in the body, different cell lines that make up our tissues and organs. Each one of those cells has a whole full genome, okay? So here we see the reason why DNA is super compacted, it's super coiled, meaning that it's shut down, functionally it's shut down. Most of the DNA is shut down most of the time in most of the cells of the body. And that's a good thing, because if it weren't, we'd have all these genes expressing all the time. And we would have, let's say, brain genes expressing in the liver, and we would have toe genes expressing in the brain, and we would have, eye genes expressing on the fingers, and we would have finger genes expressing in the heart, and that would be a royal mess, right? That's non-functional, that's non-living, that would be chaotic. So that's why most of the DNA is shut down most of the time in most cells, most functional cells. And only a little bit of fraction of the DNA opens up for expression, meaning for transcription, in that particular cell, that specialized cell. So that, let's say, in the liver cells, only the liver region of the DNA expresses. And in the brain cells, only the brain region of DNA expresses. But that's a tiny fraction. Keep in mind, of the 3% of the DNA, that codes for the 220 different cell lines that we have. And each cell line has a whole thousands of proteins that are specific, to those pathways. You can see the layers of complexity involved in metabolism. And again, it works. All we need to do is breathe and eat and sleep every now and then. And it works. Okay? In other words, we're functionally alive. Now, look at mitosis a little bit in more detail, because mitosis will set the basis for us to understand meiosis. And it's really meiosis, the goal between this lecture and next week's lecture, okay? Meiosis. So, but before we'll get to meiosis, which is a double split, we look at the single split first, which is mitosis. And these are the characteristics. It's only one cell split, so if it's one cell splitting or dividing, from one cell, how many cells do we get? 2. It's so simple. From one cell we get 2 because the cell has divided into 2, all right? Each cell. But the interesting thing is that with this duplication, the diploid number is maintained. What do we mean by diploid number? Diploid number is a reference to 2. You see the DI? DI is a reference to duo, to two, okay? In other words, pairs. The diploid number is the paired number of chromosomes. Diploid number is the paired number of chromosomes. In the human, what is that number? 23 pairs, okay? And we write it in notation, we write 2n. So 2n represents the diploid number of chromosomes in whatever species, 2n, where n represents number. Simple as that, n for number. So what is the n number of chromosomes in humans? What is the n number of chromosomes in humans? 23. 23 different chromosomes, right? What is the 2n number of chromosomes in humans? 46, because it's 2 times 23. So it's a little algebra. It's a little algebra. You'll be amazed how many people get confused with this, all right? But it's simple little algebra. N represents the number of chromosomes, number of different chromosomes, different size chromosomes in the human. We have 23, from the largest to the smallest, okay? And they're literally numbered by size. So chromosome 1 is the largest one, physically. Looking under the microscope, the biggest one. That's what it's called, a karyotype. And you've seen these images, you may not know it as a karyotype, but when we see these things, that's what we're looking at. Okay? Give me one with numbers. Here we go. Right? See? That's bigger than this guy. Okay? And this is a karyotype. One, these are the pairs, and this happens to be human. And is this a male or a female? XY, male, because the 23rd pair is not homologous, it's not identical. If it were female, it would be XX, all right? So the Y chromosome is the one that determines the sex, see?
How come they don't join at the center?
Because these are distinct chromosomes, these are chromosome pairs. It's a good question because it means that for these cells, they have already reproduced, they have already split, okay? They have split already. But the pair that you're talking about is before the cell splits. It's before the cell splits, all right? So each one of these individual chromosomes used to be a pair before they split. But let's say, to really answer the question, that this karyotype, this image, came from one of the daughter cells. The other daughter cell would have the same mapping, all right? But this is after the cell has already split, and that's why those pairs have been separated already.
So the mother cell doesn't count?
Well, the mother cell is the daughter cell of its mother, right?
Right.
Or the grandmother of the others. Yeah, you can either go up or down, either way. But there's always a mother and two daughters. The two daughters eventually become mothers when they get bigger. And that's how it is. So you can either go up or down the ladder, whichever way you want to go. Or the generations rather than the ladder. But each generation starts as daughters and eventually become mothers, unless they go apoptosis. All right? Okay. So this is karyotype. It is for the human because they're 23 and it's a male, so we can say a lot about this. And they are in pairs. So the genotype is 23, and the genome is 23, and the genotype is 23 pairs. So, all right. Yeah. Okay. So in order for the daughter cells to maintain the same diploid number, that duplication had to occur before the split. And the duplication is called precisely their sister chromatids. There's each one of those 4 legs, they're called sister chromatids. So that's another little term here. It will become more clear with a diagram. Here's a cell and interface. Cell and interface, chromatin, the single spaghetti in there. all wound up, super compacted, right? Now, the cell is undergoing mitosis. So the first thing that happens, the first stage, is the duplication. All right? Now, how many chromosomes are in this particular cell? In other words, this spaghetti was cut up into how many pieces? No. A, B, C, D. This spaghetti was cut up into 4 pieces. It was more precisely cut up into 2 pairs of pieces. There was a short pair, which is A and B, and there was a long pair, which was C and D. So again, I have a spaghetti. I cut it into 4 parts, but I'm not cutting, the 4 parts are not equal. The first 2, I'm going to make a cut, let's say for a simple number, let's say I have a spaghetti that measures 1 meter, all right? I'm going to cut 80 centimeters. 80 centimeters, and I'm going to, then what do I have left? 20 centimeters, right? So I got 1 of 80 and 1 of 20, right? I'm going to cut again. The 80 I'm going to cut in half. So I have 2 segments of how much? 40. And the 20 I'm going to cut in half again. So now I have 2 segments of 10. How many segments of spaghetti do I have now? I have 4 pieces of spaghetti. 2 of them are 40 centimeters long, and 2 of them are 10 centimeters long, right? So I got 2 pairs. I got a 40 centimeter pair, and I got a 10 centimeter pair. That's what's happened here. This particular species, whatever species it is, has 4 chromosomes. It happens that the Drosophila fly, the fruit fly, right, has 4 chromosomes. So there you go, 2 pairs. It's a short pair and a long pair, all right? Not only that, these chromosomes, so let's concentrate on the first 40 centimeters, just one of the 4 spaghetti segments, the first one, all right? So 40 centimeters, what's that 40 centimeter going to do? So the 2 daughter cells in the end, I need to end up with, again, 1 meter of chromatin, right? In the 2 daughter cells, which is going to be 40 plus 40 plus 10 plus 10. All right, on both daughter cells. But the mother cell only has 1 set of 40, 40, 10, 10. So what needs to happen for each one of the daughter cells to have the same meter? That original chromosome needs, that original chromatin needs to duplicate. All right, the duplication, which we call scientifically replication. So replication has to occur before the split. Replication has to occur before the split. This is what happens here precisely. And the first stage, there are 4 phases of mitosis. And the first stage is called prophase, all right? There's a split, the duplication. And here is illustrated the duplication, because each one of these chromosomes, you can see that has 4 legs. That's the replication there already. Replication has already occurred. It doesn't show it. Replication has already occurred, all right? So you got 2 pairs of chromosomes which are already duplicated or replicated. So for example, either one of them, C, for example, C has already doubled up. C has 4 legs, right? Let's say that these are short legs and these are long legs. The 2 short legs are identical to each other, essentially identical. And the 2 long legs are essentially identical to each other. All right, because it's a replication, it's a duplication. But they stay together, and they're held together by the centromere. It's a little white dot represented here. The next phase, they line up at the equator of the cell. Okay, they line up at the equator of the cell individually. But each chromosome is already duplicated. Then the third stage, the centromere gives way. The little white dot, this shouldn't have a white dot anymore. The centromere gives way, and the duplicates go apart, go to the opposite poles. The duplicates go to the opposite poles. That's the separation process, okay? And the fourth stage, as they go to the opposite poles, they cluster together. A new membrane, nuclear membrane, is synthesized around them to envelope them, to cover them. So we have, in fact, two new nuclei. And the chromosomes inside of those two nuclei is the same as the original mother. They should have put here, the diagram to be even more accurate, should have had an intermediate where that DNA gets cut up before the duplication so that they look like this. So the nucleus look like this, right? And without the little white dot. And then finally, very finally, after the two nuclei have configured and are structurally, have structural integrity with the DNA inside, then finally the cell itself, the cytoplasm, pinches in the middle, and that's called cytokinesis, right? Pinches in the middle and then splits, finally seals the pinch to the two new daughter cells. And that's the telos. Telos is a reference to the end. The end or the finish is the telos, is in Greek. So prophase, metaphase, anaphase, and telophase. Tell you a little anecdote. When I was in high school in Mexico City in the 1960s, all right, last century, and I was learning this for the first time. Of course, it was in Spanish, and the professor had a little mnemonic to remember, for us to remember. It was like an athlete, and it was a boys' school. It was with the Jesuits, all guys in there, and it says, this is a professional athlete, is a professional runner, so he's a pro, he's a professional runner, and he's at the starting line, la meta is the finish line, okay? So the professional athlete runs to the finish line, which is meta, is a finish line, and then he wins the race. So Anna comes over with a TV and gives him with a TV. So Anna gives him a televisor, a television, and that's a telophase. So the pro athlete got to the finish line, and Anna gifted him with a television. I'm not kidding you, that was about 50 years ago. That was a good mnemonic. I still remember it. Yeah, but it only works in Spanish, it's a problem. All right, so now what happens is, when we shift to...
Father, and after the telophase, they like wind up again in a chromatin?
Yes, exactly, thank you. What happens here is these 4 chromosomes will join at the ends, at the proper place, and match up and seal in to form the chromatin again, all right? And the functional chromatin. So there's some rearrangement that occurs here too. And it has to work well, perfectly, every time, otherwise mutations are introduced. All right, I'm gonna stop at the end of this because it's getting close to 12:45. Look at this little diagram that shows it step by step so that you can see the duplication and the distribution, how it happens. From interface, forget about those letters and let's name them like this. Now I have named them in pairs. So this type of nomenclature is more informative. This is focusing on the pairs. There's chromosome pair 1. So 1A and 1B are pairs. You can see they're bigger than 2, right? Yeah. But there are 2 distinct chromosomes, chromosome 1A and 1B. And we have the same chromosome 2 pair, 2A and 2B. In addition to that, because it's shown in an X form, in an X shape, each one of these 4 chromosomes has already replicated. Have replicated, but they have not separated. So temporarily, this stage has actually 8 chromosomes, or 8 chromatids, has 8 chromatids. See, because Chromosome 1b has two chromatids, has one chromatid and has a sister chromatid next to it, attached by the centromere. Chromosome 1a has one chromatid on this side and the other chromatid on the other side. So this is also a chromatid pair held by the centromere. And the same argument can be made for each one of the 4 chromosomes or 2 pairs or 8 chromatids, okay? Because duplication, replication has already occurred. I mentioned this because it's informative, because what migrates to the poles are the chromatids, the sister chromatids. So one sister goes to one pole and the other sister goes to the other pole. All right, just making a little more space here at the bottom to show the detail. Here's interphase, before and after. Before what? Before replication. After replication, you see the sister chromatids. All right, so one chromosome duplicates into sister chromatids, and that's what's represented in 1A, 1B, 2A, and 2B, before and after replication. During interphase is the last stage of the interphase, if you will. Then, right, sister chromatids, we get all that. Now we go into the cell cycle, all right? It happens in G2. You don't have to worry too much about the cycle itself. Here's where the synthesis, you see the single chromosomes here. Now you see the sister chromatids, the pairing up. So replication happens in this S phase, which is called S for synthesis. DNA synthesis is the same as replication. G is growth 1. G1 is growth 1, then synthesis, growth 2, and then the cell goes into reproduction proper here, the 4 stages of mitosis. But that's just with regards to the cell cycle, because you see this diagram all over the place to know that G is either growth or also gap 1, gap 2, all right? GO, no gap, is when the cell is metabolizing and the cell is just doing its function. It's not in reproductive mode. It's not in reproductive mode. I will stop here with this last slide just to set up the basis for next week. In mitosis, how many splits do we have? From one mother cell, 2 daughter cells. So we have 1 split. There's 1 split. The cell, the mother cell splits once into 2 daughter cells, right? So single split. And the daughter cells maintain diploidy, right? They continue to be diploid because of the previous duplication, so that the diploidy is maintained in the daughter cells in mitosis. And there's no recombination, which is another element that I'm introducing, recombination between the sister chromatids. In meiosis, meiosis is a little more complicated. It's like a double mitosis. It's like a double mitosis with a kink. In other words, instead of 1 split, there's going to be 2 splits, sequential. So it's going to be 1 mother cell, 2 daughter cells, and 4 granddaughter cells. Okay, it's going to be a double split. It's going to be 2 generations of cell instead of just 1 generation. And also, simultaneously, the granddaughter cells, the 4 granddaughter cells, will become haploid. They will lose the duplicates, all right? They will lose the duplicates, and now we only have single chromosomes. So those granddaughter cells are haploid, HA meaning mono, meaning single, no diploids, no duplicate. And those are going to be the gametes. Those are going to be the gametes. And so that's what happens in oogenesis and gametogenesis, spermatogenesis. That's what happens in ovaries and testes to produce the gametes, the sex cells. All right? And that is the process of meiosis, which I'm just setting up the basis now. It's a double split. We end up with 4 daughter cells instead of 2, 4 daughter cells or granddaughter cells instead of 2. And the number of chromosomes is going to be haploid instead of diploid, meaning unpaired chromosomes. Just the 23 straight up, 23 chromosomes. Okay, stop there. Torture you enough for one day. We've gone fairly deep into the thing. Just briefly to recap, in nature, inheritance is passed on in a very biochemical, mechanical, physical, physiological way without any magic involved. It is passed on by reproduction, and there are two modes of reproduction in nature. One is asexual, the other one is sexual. And asexual is a single individual splitting one way or another, different varieties of ways, into two that are essentially identical to the original individual. And so they're all different varieties of clones. The DNA in the daughters are essentially the same DNA as the mother individual. That's for asexual reproduction. Sexual reproduction, which was beginning to get into, is going to be different, and it's going to involve mixing, recombination, which allows for variation. And that's the big advantage of the double investment of energy for sexual reproduction, is that it introduces variation at two different levels, at the level of recombination of chromosomes and at the level of the mix of the specific gametes that come together. All right, stop there for sure. That's it. Thank you very much, and God willing, see you next class.